This is a production of Cornell University.
Well, thanks a lot, Matt. I really appreciate hosting, and I enjoyed having breakfast with you and Christine. And so thanks. I know it takes a lot of work to, host a person. And also want to thank Bob and Kirsten in the Soil Health Lab, who spent all morning with me showing me all the different labs. And so I've been sending samples into the Soil Health Lab since 2006, and it was neat to see where they all go. And so, thanks for taking that time. In terms of getting started here, I wanted to tell you a little story of just kind of the background and some of the motivations and inspirations that got me and my colleagues into working with cover crop mixtures. So this is a field day that I attended back in 2010, and I wasn't really sure what I was getting into when I went to this field day, but you can see the excitement, the throngs of people. It's like, what's going on inside this tent when I arrived? It's like, what are they all here for? It's a cover crop field day. And inside that tent was this guy. Okay, this is Ray Archuleta with NRCS. And he's about to do a slake test, right? He's got this soil sample here. He's going to drop it in this jar. How many of you ever seen Ray Archuleta do a slake test? Okay, a couple. You've probably seen Bob Schindelbeck do some slake tests, right? So I forget exactly what he's about to say, but it might have been, this is the soil where we grew Ray's crazy mix, and this is the soil that was left fallow and tilled, and he's going to drop them in, and the Ray's crazy mix clod is going to stay intact, and the other one's going to dissolve and erode away and whatnot. And Ray's crazy mix is a real thing. You can Google it. There's a winter mix and a summer mix. And so, then he'd go on to say, these cover crop mixtures, they're going to allow you to grow crops without any fertilizer, and you're going to be able to do no-till without any herbicides, and you're going to be able to sell the sunflowers and make an extra 30 grand per year at the farmer's market, and they're going to solve all your problems. And what's great is that it's getting the farmers enthusiastic about it, and it's getting them to plant cover crops. And I'm kind of using Ray as a prop here. He's one of the people I think has done the most to get farmers enthusiastic about soil health and cover cropping. So I appreciate his work. But what I think we need to do, and what we've tried to do over the last 10 years, is let's go from Ray's crazy mix, where it's just throw a bunch of things together and expect all these miracles to happen. So let's go to Ray's smart mix, or Ray's precise mix, where we know exactly what we're doing, why we're doing it, we're assembling the right species, we're planting it at the right time, we're measuring and monitoring things so that we know how to react in our next cash crops. And let's put the same amount of thought and precision into managing our cover crops that we put into managing our cash crops. So that's kind of been the motivation, and what I want to share today is some work that a large team of people at Penn State have contributed to over the last 10 years. So, this is a list of students and farmers and faculty members and postdocs and technicians that have all worked on multiple different grants to help improve our understanding of cover crop management and the multiple services that they provide, and how we can be more precise. And so I'm going to share work that all of these people have contributed to. I've worked in partnership with them. By no means is any of this exclusively my work. As an extension specialist, I get the honor of kind of being the front person for a lot of this work and going out and telling people about it. And so, but I just want to recognize all the people in the background of this. So one of our first questions was about cover crop multifunctionality. So the idea of mixing species together, that you could get more than you can get from an individual species alone. We wanted to test that idea and figure out, what is it about mixtures? How do you assemble mixtures that can be more multifunctional? So there's a lot of different services, biomass production, nitrogen retention, nitrogen supply, cash crop productivity, profitability, pest suppression, weed suppression, improving soil carbon, all of these services. We'd like to find a mixture that performs well at all of these as compared to individual monocultures. And so we designed an experiment at the research station initially, and also over time kind of spreading out to multiple on-farm experiments. And we had 6 species of cover crops that we tested in monoculture. So we had 2 legumes, red, and ultimately we switched to crimson clover later in the experiment, and Austrian winter pea. We had 2 brassicas, canola and forage radish, and then 2 grasses. We started with cereal rye and triticale. Ultimately, we moved to it in the second half of the experiment, years 3 through 6, and then oats. And so these are different functional groups, kind of families, plant families that have different attributes. The legumes are nitrogen fixers. The brassicas have interesting tissue chemistry and broad leaves. The grasses are nitrogen scavengers and have fibrous root systems. They're also kind of crossed here or categorized into winter hardy and winter killed species, so different growth periods and growth phenologies. And then, so growing these in monoculture, we also wanted to compare them in mixtures. And so we assembled, kind of strategically assembled some mixtures, 2 3-species mixtures. The 3-species N, we're targeting nitrogen services, so nitrogen retention by including a winter hardy grass, the rye, but then nitrogen fixing with 2 legumes. So we're trying to balance these 2 nitrogen services, achieve both of those functions. The 3-species W, which is for weeds, where we wanted to focus on weed suppression. So we have a very fast-growing fall grass, the oat, to create a canopy quickly. We have the rye to continue suppressing into the spring. But because in this experiment we're rotating into corn, we didn't really want to have a huge nitrogen penalty from lots of immobilization. So we threw in a clover as well to just kind of broaden things out. And then we started adding more species. So the 4-species mix is really similar to the 3-species nitrogen, except we threw in canola. And so canola is going to be winter hardy. It produces flowers very early in the spring. So maybe we increase the service provisioning to attracting beneficial insects or resources for native pollinators. Also, interesting tissue chemistry with canola having the glucosinolates. Maybe that's going to affect the microbiome or do something for weed suppressiveness, something like that. And then finally, all 6 species together, kind of the more is better. Insurance hypotheses, maybe we have some redundancies in functional traits in here, but also a highest species richness. So one of the ways to measure diversity is just pure number of species. So this would be the richest of the mixtures. It's planted on a 30-acre long-term site. So this square of plots here is the 30 acres. It's certified organic. It's in a 3-year crop rotation of winter wheat. And then after the winter wheat is harvested in July, in August, we plant the cover crop treatment. So the strip here where you see the multiple plots of different colors, this picture was probably taken in September. And so you see our different cover crop treatments down the length here. So that's all the monocultures plus the mixtures and a fallow. That cover crop will then get terminated in the spring and go into corn, which I believe this is our corn crop right here. And then after corn, we just plant a monoculture of cereal rye and then rotate into soybeans is the 3rd crop in the rotation. Then the soybeans get planted into wheat. So it's an annual rotation organic management in this experiment. So when we looked at the multifunctionality score, so if you take all of those services and kind of standardize them to the mean value of that service and then average those mean values together, we get a average multifunctionality score. If we just want to think about richness, so if a farmer wants to get multifunctionality and they just think, okay, should I have 3 species, 4 species, 6 species, is increasing the species number, is that what's going to take to increase multifunctionality? What we saw is a very, very weak relationship to average multifunctionality. Some of our monocultures, like the pea, were even more multifunctional than any of the mixtures. We've got some variability around the line here, but in general, 3 versus 4 versus 6, not that different in terms of their multifunctionality. So it seemed like just increasing species number is not necessarily the key that farmers should use as their strategy for assembling a multifunctional cover crop. We looked at a number of other metrics, diversity metrics, and we started getting into the functional diversity metrics. And I'm going to talk a little bit about functional diversity in a second here, but the one functional diversity metric that was the best predictor of multifunctionality is this term called Rao's quadratic entropy. How many of you have heard of or know of Rao's quadratic entropy? Anyone? Okay, I'm not surprised. Very, very few people are as into the weeds to know what this is, and it's certainly not listed on a cover crop bag of seed, right? You know, you're not going to get the percent Rao's quadratic entropy. You know, you might get the amount of weed seeds in there, but you're not going to get the, you know, tested by the Department of Agriculture, but you're not going to get Rao's quadratic entropy. So what is Rao's quadratic entropy? What is functional diversity in general? So you need to understand functional diversity before you can understand how to make a cover crop multifunctional. So functional diversity has to do with not necessarily the number of species, but what the functional traits of the species are. And when we talk about functional trait, this is kind of like a physiological trait or of a species. And so things like fall growth rate. So how many pounds per acre per growing degree day will a species be able to put on under optimal growing conditions? And these functional traits are related to the services of interest. So for example, fall growth rate should theoretically assist with weed suppression, being able to retain nitrogen, being able to produce biomass of that cover crop. Spring growth rate should help with nitrogen retention, carbon inputs. The C to N ratio should be able to help with nitrogen supply. The list of functional traits could go on and on and on and on. You know, root length density, leaf area index, flower density. There's endless list of functional traits. We selected these because they're things that agronomically are easy to interpret and easy to measure, but you know, the list could really be expanded. And I think some of the future of multifunctionality in cover crops is what are the additional functional traits that we could measure and use to predict different services. So that's what functional traits are. But then to understand what functional diversity is, you have to think about these multiple different traits and how they arrange themselves in what we call the trait space. And so this is with 3 traits, it's nice and easy to be able to, you know, plot things on a 3-dimensional axis and, you know, visualize this in space. But mathematically speaking, you know, we can do the math on, you know, more than 3 dimensions, 5 dimensions, 10 dimensions, 12 dimensions of trait space. The math all works for it. But essentially, you know, thinking about an individual species, where it falls in the trait space. So for example, red clover has a relatively low fall growth rate, a kind of medium spring growth rate, and a very low C to N ratio. Rye has medium fall growth rate, but very high spring growth rate and a high C to N ratio. So that's where it falls in this 3-dimensional trait space here. So then when you start assembling mixtures, you can see where do the different species in each mix fall in this 3-dimensional trait space. So here's our 3 species N that's got the rye, the pea, and the red clover. So that's what the functional trait space of this mixture looks like. For the 3 species weeds, you know, again, kind of a triangular plane there of trait space. We add the 4 species here. We've got canola in the middle. So I've kind of created this convex hull around them with the lines here, and then our 6 species mix is here. So this is each of the mixes in the trait space. The next thing with functional diversity is understanding components of the functional diversity. So you can have components of diversity, whether it's based on functions or based on just species numbers. There's evenness, richness, and divergence. And in evenness, in functional diversity, evenness would be a perfectly even mixture would have the same amount of biomass of each of the species in the mix, right? third of the biomass with this, third, a third, or quarter, 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 or 1/6, 1/6, 1/6. That would be a perfectly even mix. The more you have biomass concentrated on one species versus the others, the less even you get, right? So that's kind of, pretty simple one to think about. Richness, if we're thinking about kind of species number, richness just means increasing number of species. But when we're talking about functional diversity, what richness is an increasing volume in the functional trait space. So if you were to draw this convex hull around the species in the trait space, the bigger that volume is, the richer your functional diversity is. So here, if we go from, say, the 3 species N to adding canola, we've kind of taken a three-dimensional plane and we've added another species, and so now we've got kind of a pyramid. So we've increased the volume. Here we've got more species that are increasing the volume, the size of that hull. That's a more, that's a richer, functionally richer mixture. And then the last thing we can look at in terms of the diversity component is divergence. And divergence means how far apart from the middle are your species spread. And so these would be very divergent. The 3 species mixes here are very divergent because all of the species are at the edges of the trait space. When we go from the 3 species N to adding canola, which is kind of in the middle of the trait space, we actually reduce the divergence because we're bringing weight or emphasis into the middle of the functional trait space rather than keeping it at the edges of the trait space. So there's lots of different metrics that you can calculate, metrics like relative Rao's quadratic entropy, that will kind of weight on evenness or weight on richness or weight on divergence or kind of combine evenness and richness together or look at all 3. And relative Rao is a metric that essentially measures the evenness and the divergence of the functional trait space. And so what that's telling us, the fact that relative Rao, of all of the possible metrics we could look at, relative Rao being predictive, and this is the second paper where we found relative Rao was most predictive, so 2 different data sets. So it's a little bit robust in that regard. It's the evenness and the divergence. And so what that tells us is we want to have even species composition, and we want to have species that are on the edges of the trait space. So, for example, putting some emphasis on canola, which is in the middle of the trait space, that's actually going to reduce our divergence. What's interesting is richness didn't really matter. It doesn't matter how far out or how big your volume is, as long as you've got some strength around the edges and it's evenly distributed. That seems to make for a more multifunctional mixture. So I think this helps us understand better what the functional aspects are and how we can manage for them. Now, the other thing I want to say is that the functional traits that you throw into this analysis could be in the eye of the beholder, right? We don't have flower density on here. Canola has huge flower density. If that was one of our functional traits that we were interested in for bringing in pollinators, canola wouldn't be in the middle anymore. It would be way out here. Okay, it would now be on the edge. So it's really a little bit subjective about what your goals are. So I think there's an opportunity for farmers to be able to say, okay, I want to have flowers. I want to have a dense root system to build aggregate stability. So maybe root length density would be one of my functional traits. I'm not that concerned about nitrogen supply. Maybe I've got a lot of manure to utilize. So maybe I'm not going to throw C to N ratio in as one of my traits of interest. So I think this is also a framework that's adaptable and can help farmers, hone in on what's important to them and then have a quantitative framework to help assess, okay, these are the ones that are going to be my divergent species in this trait space, and then we're going to try and manage them for evenness. So one of the challenges, though, is the evenness aspect. And in the first round of our experiment, we had a lot of challenge developing even species distributions over time. So this chart shows kind of over the progression of the cover crop from seeding to germination to fall biomass to spring biomass. What is the proportion allocated to each of the species? And you can see over time, this green bar here, which is the cereal rye, tended to dominate things in the spring. And so that's a challenge because that's going to disrupt the evenness of the mixture there. So this is a picture of the cereal rye in the spring. Cereal rye is just, those of you who worked with it know it's very fast growing in the spring. It got way out ahead of all of our, peas and red clover, and it's very uneven. On other settings, the canola became dominant. So this is one of our on-farm locations in a warmer part of the state. 4-species mix just turned into all canola, right? So these are some challenges that, you have the best intentions, you make this nice seed blend, and then the environment kind of swamps everything. So how do we look at what the environmental context is and adjust and work around that? Just some examples here of our 4-species mix that we put out across 4 different on-farm locations and our research station, and it behaved very differently depending on the context. The yellow part of the pie being the proportion of biomass that's in canola. Canola really responds like crazy. So, we had this question of how do these cover crop mixtures respond to environmental conditions over different space, which might be different climate regions or fertility levels in the soil. We had a hypothesis that the planting date and the nitrogen rate would potentially control mixture expression. And so we set up an experiment at our research station, which this is a picture of it here, where we experimentally manipulated the planting dates in the fall and experimentally manipulated nitrogen additions. And then we also just deployed satellite experiments on different farms around Pennsylvania. And we actually had a couple sites in New York too. This is Tor Oshner's farm up here, so not too far from here. And we grew the cover crops, we compiled all the data, and we wanted to analyze it and say, okay, let's look at our hypothesis of nitrogen supply and climate in the fall in terms of growing degree days. How does that affect the composition of this mixture? And so we used a path analysis to look at it. So kind of our independent variables here are inorganic soil N at planting. This is milligrams per kilogram of nitrate and ammonium at planting. And then the growing degree days between planting and December 31st. And you can manipulate that either through your climate. You can be in a colder region that just has fewer growing degree days, or you can manipulate that through planting date. So if you plant 3 weeks earlier, you're going to accumulate that many more growing degree days in the fall. And so what we saw is that growing degree days, increasing growing degree days, increased growth of all of our species. This is fall cover crop biomass N. And the only species that really responded to the inorganic soil N at planting was canola. And then these coefficients essentially give you an idea of, for every unit change in inorganic soil N, what's going to be the change in canola biomass in kilograms per hectare. So 1 part per million of inorganic N is going to create an additional 44 kilograms per hectare of canola. And 1 growing degree day is going to create 2.8 kilograms per hectare of canola. And one of the things that we see is that canola is much more responsive than the other species. So none of the other species responded to inorganic N, and canola responded 5 times as much as the next species, actually 6 times as much, 7 times as much as the next one, triticale, in terms of growing degree days. So an extra growing degree day for canola is much more impactful than for triticale. So canola is extremely responsive, and then these negative coefficients are kind of interspecific competition. So the canola, an increased kilogram per hectare of canola decreased the growth of all of the other species. So in this way, canola is kind of acting like a little bit of a keystone species in terms of having these effects on the community. This is what the data looks like just at the research station in terms of our different planting dates and our nitrogen additions. You can see as the planting dates got earlier, we had more growing degree days. The yellow bars for canola get bigger and bigger. And within each planting date, adding nitrogen stimulated the canola. And just a visual assessment on the first planting date, this is what the mixture looked like with canola dominating when we added nitrogen. When we didn't add nitrogen, there's much more legumes and triticale that are able to persist in the mix. So these are things that, farmers can choose their planting date in the fall. If they're coming after wheat or a small grain, they've got a very wide window where they can plant. They could choose to plant in early August, or they could choose to plant in mid-September. And it seems like if you want to get a more even composition, then planting a little bit later is important. You could also choose whether or not you're going to add fertility. So if you've got manure on your farm, say, you know what, if I've got canola in a mix, am I going to put manure out? Probably it's not a good idea, because you're just going to disrupt the composition of that mix, make it very uneven. So I think there's some management factors that can be used. The other thing would be, you really need canola in the mix, right? So maybe canola is a destabilizing force in the composition. It's so reactive and it's so competitive that maybe things would be more even and stable without that species. So I think for resilience of cover crops, it's could be an interesting research line to think about how resilient are different cover crop mixture species to different environmental factors and things like that. One of the other things we saw with planting date is the weediness too. So if you're trying to use cover crops to manage weeds in the fall or manage the weed seed bank, it seems like the earlier we plant our cover crops in the fall, the weedier they get. So this is a random forest analysis, actually. Very large data set, thousands of plots from lots of different experiments where we measured the cover crop biomass as well as the weed biomass in the fall. And as you increase the fall growing degree days here, the weed biomass, so this response variable here is weed biomass, really starts to tick up. Here's some examples from the planting date experiment where we had triticale planted in early August. You see how infested with weeds it is versus triticale planted in early September. And yeah, there's some things that germinated there, but it's cool enough that those are not really going to get out of control. They're not going to set nearly as many seeds. So this could be a weed management strategy here too in terms of the planting date. So what I want to move into now is work that kind of my lab group has really focused on over the years, looking at how we can better assess the nitrogen contribution of cover crops as well as soil organic matter to the fertility of our following cash crop. And we've really looked at corn as the model system here, because corn is it's one of the predominant crops on the landscape. It's very responsive to nitrogen to maintain yields. And so the way we wanted to start looking at this work is to start at thinking about the fundamental theory and the fundamental processes of nitrogen mineralization, and use that as a guiding principle in terms of how we interpret and analyze the data and be able to predict a response. So it's a fairly simple conceptual model of how nitrogen mineralization occurs in the soil. You've got a decomposing organic material with carbon and nitrogen. Microbes decompose that material. They might use 25 to 50% of that carbon to build their biomass. That we call that the carbon use efficiency. The remaining fraction is respired into the atmosphere as CO2. The quantity of carbon that microbes assimilate, they need to assimilate a similar or a proportional quantity of nitrogen to build their biomass, to build the proteins and amino acids and whatnot that they need. That seed-end ratio is often about 10 to 1, but it might vary if it's bacteria or fungi or protozoas. But 10 to 1 is kind of an average value that a lot of people use for the whole microbial community. And what of the nitrogen that decomposes from this material, what the microbes don't take up into their biomass is what's available to be mineralized into the soil for the crop. And of course, those of you that know this process well know that you cannot have enough nitrogen in the decomposing material to meet the microbial biomass demand, and then you immobilize nitrogen. So you could actually tie up nitrogen as the microbes build their biomass. So the cover crop seed to N ratio is an important control on this. So that's the theory. And the very nice thing about this theory is that it's pretty easy to turn it into an equation. So this is the classic biogeochemical equation for nitrogen mineralization. I've got the C to N ratio of the cover crops, the C to N ratio of the microbes, the carbon use efficiency, you've got the N content of the decomposing material here, you've got a coefficient for the decomposition rate, and that's nitrogen mineralization. So we wanted to use this equation and this theory to say, okay, we've got a cover crop residue that we can measure the nitrogen content, we can measure the C to N ratio, we've got soil organic matter that we can measure. You can measure carbon and maybe it's got a fairly stable C to N ratio. As pools that go through this process of decomposition, let's see if we can use this as an equation to predict nitrogen supply to the corn crop. And so what we did is we distilled a set of equations from the theory that could be calibrated to predict essentially the corn yield response. So we didn't look at soil mineral nitrogen, which is what a lot of people do. We said, we want to take this right to yield. We want to see how this mineralization process affects the yield of the corn. So that's our response variable, is the yield of an unfertilized corn crop. We have a parameter for the carbon use efficiency, or actually I call it the humification efficiency, and humification is a little bit different than carbon use efficiency. Carbon use efficiency is for one generation of microbes, but you've got a 3 or 4 month growing season where you might have multiple generations of microbes. And so you need to account for, okay, the bacteria are going to get eaten by protozoa, and then those protozoas are going to die. And so your carbon use efficiency might be, or your overall efficiency, which I call the humification efficiency, might be lower. That humification efficiency, there's also a lot of theory to suggest that that's affected by the soil texture. So finer textured soils with more clay content can stabilize carbon, stabilize dead microbial biomass, protect it against decay. A lot of that work has come out of Cornell. So I'm appreciative to those who have worked on that. So I've read a lot of those papers and gained a lot from that. So our efficiency factor, we said theoretically, okay, we're going to allow that to vary based on the soil texture. We've got a credit here for the yield response to soil organic matter, and that's going to be, again, affected by the humification efficiency. There's a few things that simplify out of this equation. We assume that the C to N ratio of microbial biomass and the C to N ratio of soil organic matter are going to be similar, 10 to 1. So those actually cancel out with each other. We just get 1 minus E. And then these coefficients that you see in here are coefficients that are calibrated to our data set. So I'm not doing any hand waving here. It's just the data is telling us what these should be. We've got a yield credit for cover crop residues decomposing. So this is our, kind of full formalized equation here. We've got the N content of the cover crops, the humification efficiency, the C to N ratio of the cover crops, and then alpha is a parameter here which composites both the decomposition rate as well as the efficiency of that mineralized nitrogen in converting into corn yield. Okay, so our response variable is always the yield. So this kind of takes into some nitrogen use efficiency as well. And we found that parameter depended on whether the cover crop was going to mineralize nitrogen or immobilize. And when it immobilizes, this value becomes negative, and actually the effect of an immobilizing cover crop is much more strong than the effect of a mineralizing cover crop. And that's interesting, because that nitrogen mineralization, some of that mineralized nitrogen might get lost, might get denitrified, it might get leached. And so that's kind of an interesting finding. And then the last thing we said is, okay, we've got this credit from the organic matter, credit from the cover crop. Let's add these together. And we know that corn yield kind of has a diminishing return to increased nitrogen supply. And so let's throw a quadratic term in here to represent that diminishing supply. So this was our system of equations. And we threw a data set at it from 125 different plots that were grown in different sites with different textures, different soil organic matter levels, different cover crop residues, different nitrogen levels. And we said, let's see if we can calibrate these terms. And we were able to do that, and we got a pretty good R squared. And we had a wide range of unfertilized yield potential across all of these different sites. So I think it's a big advance in terms of being able to very precisely and with very strong biogeochemical theory behind it, be able to predict the nitrogen content or nitrogen contribution of soil organic matter in cover crops to the yield of corn. So now that we've got this out of the way, the next step is figuring out how do you take that to a nitrogen fertilizer requirement. And that's kind of a giant leap in and of itself. And we're in the process of making that leap right now. But the approach we're taking is, if you can predict what the yield of your crop would be without fertilizer, and you know from history what you've been able to achieve through abundant or possibly overabundant nitrogen applications, you've kind of got a yield goal in mind for your particular climate and soil and management system. And so the difference between your yield goal and what you can get without fertilizer, we're calling that the yield gap. And that yield gap, to produce a bushel of corn, physiologically, the plant needs to take up about 1.2 pounds of nitrogen to produce a bushel of corn based on the stoichiometric ratios of carbon and nitrogen in a corn crop. But we know that that nitrogen isn't always delivered with 100% efficiency. So you've got to adjust by some efficiency factor. And a lot of our research now is kind of trying to figure out how do we test what this efficiency factor is and how do we predict that or adjust for that. And so, in the future, we're going to have, some results to be able to share on that. But for now, I'm going to kind of leave the story of developing these algorithms here. The next step, though, is to kind of create some user interfaces. Like, that's a nasty system of equations to have a farmer sit down, and I have created a worksheet. It's just multiplication, division, and addition. You can do it, but it's, some people look at you funny when you ask them to fill out all that things. It's like you're doing taxes or something. So we put it in a web interface to do all the equations, and the interface predicts your unfertilized yield, and you can plug in your yield goal, and there's a yield gap there. And we're right now kind of making some assumptions about what the fertilizer efficiency is, but we're trying to fine tune that. And so this is a tool that kind of field testing with some select group of growers to get their reactions and see how accurate it is and do some fine tuning. One of the other advances that we've done to try and make this more practical and easier for farmers to do is, instead of taking your cover crop biomass and going out there with a quadrad and getting down and clipping it and drying it and sending it to the lab, it'd be much nicer if you could just walk a sensor over that field and get the nitrogen content. So there's an NDVI sensor here made by GreenSeeker that's a handheld sensor that is $500, and you can walk that across a cover crop plot. And I've just kind of on a hunch, I got one of those back when it was first released, maybe 2012. And every single cover crop experiment we did, I said, all right, let's walk this across the cover crop plot. And we were able to develop a really nice relationship between the NDVI value on the sensor and the above ground biomass and content. And I think it's accurate enough, and we've done some sensitivity analyses in our equations that I think even with some error, it doesn't really change the outcome too much. And so I think this is going to be a nice tool going forward that, if you need that nitrogen content to run some of these predictions, you can get that with a sensor. You can also get a model of this GreenSeeker sensor that you can map a field. So you could put it on a spray boom or a toolbar of a tillage implement. So if you're going across that field, you can get the NDVI value and make a map and adjust spatially. So one of our projects that we've been doing the last 2 summers has been to look at in-field variability of the cover crop nitrogen content. So got 2 GreenSeeker sensors here on a little boom here on our buggy, and we drive this across the field in swaths. We're pulling behind it a sensor that maps the electrical conductivity of the soil, which is a surrogate for soil moisture, soil texture. So, that texture component, we can find different zones in the field and, create prescriptions for that. This is an example of one of the cover crop fields that we mapped this spring. You see there's a really wide range from 15 pounds of N to 135 pounds of N in the same field. So be able to account for some of that variability, you could adjust what the nitrogen credit is. It's a picture of this field and us driving the buggy through it. I also think there's potential to use satellite imagery. So most of the agronomists I work with now are either, have subscriptions to either Encirca through Granular or Climate Field View to get daily satellite imagery of all their fields as long as there isn't cloud cover to their smartphones. And this satellite imagery map looks a heck of a lot like this NDVI map. It'd be nice if you could just get that for all your fields right to your phone or download it into your workflow and create some prescriptions using that. Map of the soil electrical conductivity zones for this field, can then go out and soil sample by zone. And a lot of farmers have already invested in these EC maps and have developed, management zones or soil sampling zones. So they're already doing this. And so let's just get the soil texture and the organic matter from those zones, and we can layer that in. Corn yield potential by zone. This is, historical records from this farmer, and he's he's pretty savvy. He's done all the work himself to create these yield zones that he already uses to manage his P and K applications to account for varying levels of crop removal. And then we put all that together. We run the equations I showed you by zone, and we get a nice map of maybe what the nitrogen fertilizer recommendation should be by zone. So this is still all very experimental. There's kinks that we need to work out. That efficiency factor I showed you for the nitrogen, that's a big unknown that we're trying to hone in on still. But kind of a proof of concept that you can layer these things together and maybe be more precise about how you credit the nitrogen from cover crops. So, it's been 10 years since, I went to this field day. It, inspired our group at Penn State to really look into this. This is what a field day for hemp production looks like now. So, kind of past the peak for cover crop mixtures. I think we've learned a lot. Farmers are kind of over the adoption curve. They're doing it now. It's time to get smart about it and precise about it. The cover crops can provide a lot of benefits, but they need management. They need intentionality. So you think about functional diversity and the evenness and the divergence as guiding principles. Adaptively managing cover crops to promote evenness, so avoiding suppressive species like canola, minimizing residual nitrogen, not planting too early, and then some decision support tools for managing cover crop nitrogen is something that we've worked a lot on in my group and are trying to move forward. So I'll leave it at that. Hopefully there's a few minutes for some questions and discussion. So thank you. Yes, sir.
So the one decision tool you talked about was focused on looking at nitrogen after the cover crop. Have you done that in the opposite direction? Say we have this goal for nitrogen, what should we use in our mix? And then maybe beyond that, have you looked at other goals for the farmer, like weed suppression, and how that can be put into a decision tool that sort of integrates what different goals and different climatic factors might be influencing their cover crop decision?
Yeah, so I'll just repeat this in case people online. So the question is, in terms of decision support on the nitrogen side, have we looked at the reverse of saying, let's run some scenarios and figure out what the right cover crop is in order to get a certain amount of nitrogen, and then also for like weed management or other services, are there decision support tools? On the nitrogen side, we definitely can do what you're describing. We haven't advanced enough to work with growers to use it in a proactive way. But the decision support tool I showed you allows you to run scenarios. So let's say, dial in your organic matter, dial in your soil texture, dial in your yield goal, and then you can adjust the amount of cover crop nitrogen, you can adjust the C to N ratio and see what that does to your unfertilized yield and say, okay, you know what, we're really close to our yield goal here without any additional fertilizer. We don't need a massive legume cover crop. Maybe we could go with a mixture or a cereal, something like that. Or if we're really far away from our yield goal, we're going to need a big, massive legume monoculture here. Let's focus on that. So yeah, it definitely can be used that way. On the other side, in terms of other additional services, I think it would be great. I think with a quantitative framework of kind of functional diversity, I think there's an opportunity for someone to create that. It's a little bit beyond the scope of what I'm looking at in my group now, but I think maybe others, maybe the Cover Crop Council. I think the Cover Crop Council is working on something like that will be able to, allow growers to check off some functions that they want and then help design a mix there. Yep. Yes, sir.
On those predictions that you're making, the contribution of nitrogen from soil organic matter and from cover crops, those equations, I didn't see any mention of like soil temperature, soil moisture, or maybe flooding. Do you include that, or is it something that you can leave out?
Yeah, so the question was, there was about the nitrogen equations. There's no mention of soil temperature, soil moisture, soil flooding. And right now, we do not specifically take those into account on a site-by-site basis, but through the calibration process, all of these sites were calibrated in central to southeast Pennsylvania, which has kind of an average moisture regime, average temperature regime. It's all calibrated to the average conditions, and these were done over 4 or 5 years. So it's calibrated to the average conditions of Pennsylvania, and that coefficient absorbs some of that information. Now, it's not going to be able to tell you the difference between a year that's hot and dry, or a year that's cold and wet, or a year that we have lots of denitrification. And I think this could be used as a complementary tool to some of the models like AdaptN, where you are taking into account those very specific year-by-year factors. But of course, there's a cost to AdaptN in terms of or other models like that in terms of the computer infrastructure, and requires subscription services and things like that. What I really wanted was a tool that was calibrated to the average condition that you actually could do on a piece of paper. I think that's a really powerful thing, and with some very simple inputs. That was kind of my goal. So doesn't take into account everything, but I think it works well for the average. And the other thing is that if people want to use these equations in New York or Maryland, I'm like, good luck, because these are calibrated for Pennsylvania. And we've talked with some people about, okay, let's get a calibration for Wisconsin, or let's get a calibration for Maryland. I think you could do that. You could do these calibration studies. They're not that hard. People probably already have the data actually from existing experiments. And I think those coefficients are going to change based on the temperature and moisture regimes. Yes?
Oh, thanks very much. I have two questions. One is, have you looked at canola as a crop at all? And the second question is kind of your functional space, trying to make that bigger. If you had all winners, or one winner that does all three things perfectly, that would be one dot, but it might be the best. So I'm a little confused about a function, sort of a bigger space is basically saying I want to have like failure on multiple dimensions.
Yeah. So that's a great question, and those are great points. So one of the things we saw, if you remember the species richness multifunctionality response, the monoculture P was actually the best multifunctional performer. And I think that's speaking to what you're saying of a single species that can do multiple things well, turned out to actually be the best of the multifunctionals. In terms of functional diversity, the math falls apart when you only have one species, right? Because you've got a single point.
You can have 6 that are all winners, and you still have that same single point.
Yes, I think so. I'd have to think through that. I'm not able to fully think through it on my feet here, but I think that's possible. And I think some of what our data was showing is that, yeah, you don't always need a mixture to be the most multifunctional, or to achieve multifunctionality. You can sometimes get that with the monoculture. In terms of canola as an actual crop, one of the long-term experiments on our research station that was just to the side of our cover crop mixtures experiment has used canola as a crop, and they're actually harvesting as an oil seed, and they press the oil, and they use that oil to run the tractors, and then they use the meal to, as a dairy supplement in the ration. It's interesting. In theory, there are a few farmers that are growing canola in Pennsylvania. They're kind of growing them for the human food market. It's a very specialty niche crop. It was a devil of a crop to grow. It's hard to get established. It gets eaten by slugs. There's tons of harvest losses with the combine. So not saying it can't be done, but it takes someone who really wants to grow canola to be able to do it. It's not something that we're seeing average farmers really jumping at adopting. Yep. Who's? Okay, go ahead.
Thank you very much. Very thought-provoking. I wanted to dovetail on those questions that are very interesting. Now, it strikes me that maybe there is no average here ever. And so with the multifunctionality as well as the prediction of nitrogen delivery, the robustness maybe lies in coping with a range of environmental conditions, a dry, a wet year, and everything in between and all. So by looking at average performance, both for the nitrogen delivery as well as the multifunctionality, have you thought about, or how can you mold this in a different way that actually robustness to erratic conditions, which is, I think, where the sustainability comes in, not the average that doesn't exist.
Yeah, I think that's a great point. And I think we have had some students that have started off looking, or started to look at that in terms of performance over different years and the resilience of the systems. And I'm not super familiar with their work. I'm not their advisor, and so I think people are thinking about that, and I think that's a really important point, because I do agree that the average doesn't seem to exist anymore. It's just a lot of variation around the average in any given year. So, but I do still think that so finding things that are robust and resilient, I think is important. And I think some of the work we're starting to do with our nitrogen tool is to kind of look at the sensitivity of it. So how sensitive is it to the different parameters, and is there some level of stability that maybe we could look towards? So I think those are really important points. It's a well-taken comment.
Not only in time, but also in space, right? You mentioned in space that there's also no average.
Yeah.
Right? Everything is away from the average.
Yeah. Yep. Okay. Well, thank you again. Appreciate the opportunity to visit.
This has been a production of Cornell University, on the web at cornell.edu.